And just turn it over to Andrea, who's joining us from Cornell in the Pollock lab, I think. So I'll just give it over to you, Andrea. Thanks.
Well, hi, everybody. It's great to be here. Thanks so much for coming to EYH, and I hope everybody's learning a lot and is having a good time. So let's see. Can you guys see my screen okay? All right. So today I am going to talk to you about how scientists can use X-rays and these beautiful little gemstones here to solve some really interesting and important mysteries in life and biology. But before we get into all that, I wanted to tell you guys a little bit about me and my path to becoming a scientist. So when I was in junior high and high school, I was a huge science fan. I loved all of my science classes, but in particular, I was really drawn to astronomy. And here's a picture of my dad and I with the telescope that we built. I knew that I wanted to continue learning science, so I went off to college and I majored in astrophysics. And here I was lucky enough to be able to use even larger telescopes. Now, while I was at college, I knew I wanted to be a scientist, but I decided that I wanted to switch gears and work on something a little more directly relevant, a little more useful to people here on Earth. So I became a graduate student in Lois Pollock's lab here at Cornell University. I hope some of you guys heard Professor Pollock talk this morning. So while I was a grad student here, I had these wonderful opportunities to do some really fascinating biophysical research, and I earned my PhD about 1.5 years ago. Now, this is only one path towards becoming a scientist. But some of you guys who are thinking about science careers for yourselves, you might be very curious about becoming a grad student and what that's like. And we've heard a fair amount about that today, so you guys are a bit more in the know. But I find that usually when I told people I was a graduate student, they would be kind of confused. They'd say, well, what does that mean? A lot of people would think maybe I did homework all day, but in reality, I didn't really take that many classes. So what do graduate students actually do? What's it really like? Well, that varies a little bit depending on which field you're in and which lab you're in. But in general, graduate students are focused on doing scientific research and solving important mysteries in science. Now, in the Pollock lab, we made new discoveries in biology and in biophysics. Now, some of you guys have taken biology or are taking it right now. And you might be thinking, well, wait a minute. There's all of these textbooks and there's so much information in there. Surely isn't all of biology known already? But it turns out that no, even though there's this huge volume of written knowledge, we've barely scratched the surface. There's many mysteries remaining and scientists everywhere are working to solve them. Here's an example that I really like to give. This little guy is called the Olinguito, and he's a brand new species of animal that we discovered for the first time in 2019. So clearly there's still lots out there that scientists don't know. Now, unfortunately, in my lab, we don't get to study new species of animals, but we do get to look at how living things function on a molecular level, mysteries of life on a much smaller scale. What goes on inside of this little guy at a very, very tiny scale that allows him to exist and function as the living creature that he is? So let's take a bit of a deep dive into this. What do I mean by molecular level? Well, let's start by thinking about life on a scale that we're a little bit more comfortable with. Let's start by thinking about our hands. Your hands are in front of you all the time and we're very comfortable with them. But what were to happen if I had a special magical magnifying glass that I could use to zoom in a factor of 1,000 times on a little tiny part of my hand? I would see that my hand is made up of cells, which some of you guys may have heard of in biology. Now, cells are building blocks of life, and the cells are actually really incredibly complicated. If you were to look inside the cell, you would find that it's made up of all sorts of smaller parts, things called organelles. And each organelle has its own role and function that's essential to the survival of the cell, and therefore to the survival of us, because we're made up of cells. And by the way, stick around for the cell demo after this if you want to learn a lot more about cells and organelles. Now, as complicated as this cell is, let's imagine that I were to take my magical magnifying glass again and use it to zoom in another factor of 1,000 times on one of these organelles. What would I find then? Well, I would find that the organelle is made up of even smaller things called biomolecules, things like proteins and nucleic acids. And these biomolecules make up everything in your cell, and therefore pretty much everything in our bodies. And these are what we study in the Pollock lab. So we study biomolecules. Why? Why should we care? Well, I told you that on a very small scale, we're basically made up of these biomolecules. So the biomolecules are really responsible for almost everything that happens in your body. But it's not just that. It's not just people that are made up of these biomolecules. It's things like viruses and bacteria. So biomolecules are responsible for everything that happens in them too. So given that, you can imagine that if we could study these biomolecules, we could get more insight into how our bodies work. And we might even be able to help people who are sick get better faster. So definitely good goals. We definitely should study these things. But what do I mean by study them? I mean, there's lots of things about them that I could study, right? Well, in the Pollock lab, we like to study biomolecular structure, which is basically a fancy way of saying, what do these molecules look like? Now, the idea is that if we could learn what they look like, we might be able to get some insights into how they work. And biologists have this saying that they like to use that goes, structure dictates function. Now, I used to find this kind of confusing, but I like to think about it in terms of construction vehicles. So let's imagine for a moment that you've never been to a construction site before. But you can tell just by looking at these things that they're not intended for the same purpose. And you could probably make a guess just by what each of them look like, what they're meant to do. You could probably guess that this one might be meant to dig or lift something. This one might be meant to roll over something and flatten it out. So in the absence of any other information, just knowing what things look like can give you a lot of insight into their function. And believe it or not, biomolecules are like this as well. So let me give you another example. Let me give you a more concrete example of why we care about biomolecular structure. And for this example, I'd like to talk about antibiotic resistance. Well, this is a bit of a buzzword lately, so some of you guys might have heard of it. Let's imagine that I have a bacterium right here. Now, bacteria are just single cells, just one, but that cell is made up out of biomolecules. Now, let's imagine that this is the kind of bacteria that can make you sick with strep throat. So you go to the doctor and she prescribes you an antibiotic. Now, the antibiotic works because it can bind to and interfere with the biomolecules that make up the bacteria's cell wall, and it can keep them from doing their job. Now, when that happens, the bacterium explodes and you get better, which is great. That's exactly what we want. But unfortunately, some bacteria have evolved to have a sort of workaround for this. Let's imagine that I have a new bacterium, and this one in its cell wall has an additional kind of biomolecule. And this biomolecule is capable of chewing up and destroying the antibiotic before it can go to work on its normal targets. Now, when this happens, the antibiotic is pretty much as good as garbage, and you don't get better. So obviously this is a big problem. We don't want this. We need to find a way to fix it. The challenge then becomes to create new antibiotics that don't get destroyed. And you can imagine that if we could learn what this molecule looks like, we might be able to design a better antibiotic. We might be able to design something that has the perfect shape that this biomolecule can't grab it. So I hope this example helps you see why it's important to study biomolecular structure. But how would we go about doing that anyway? How do we see these things? Can we look at them in a microscope? Well, it turns out, unfortunately, no. Here's a state-of-the-art picture of a microscope looking at cells. And this is a gorgeous image. You can see lots of detail, we can see some nuclei, but unfortunately, we can't see the individual biomolecules. So why not? Why can't we see them? Well, the first thing you need to understand is that light is a wave. It's kind of like a wave on the ocean, except it travels through the air and we can't see it. And the light is incredibly tiny. So the distance between different peaks in this wave, which we call the wavelength, that is less than a millionth of a meter. So very tiny little waves going through the air. Now, what would happen if we took a biomolecule and put it in a light wave? Well, it turns out that as tiny as the light's wavelength is, the biomolecule is way, way, way tinier. And this isn't entirely correct, but you can kind of think of it as being that, well, the biomolecule is so small that the light wave doesn't even know it's there. It's kind of like if you were to put on boxing gloves and try to pick up a grain of sand. It's just too tiny. So how do we go about seeing them then? Well, there's a bit of a trick that we can use, which is that each color of light has a different wavelength. Red light has slightly longer wavelength than purple light. And while it's true that none of these are small enough to see biomolecules, the truth is there's many colors of light that exist that we can't see. So some of you guys might have seen a chart kind of like this before. This is called the electromagnetic spectrum. And these are all different kinds of light with different wavelengths. And what we usually think of as light, visible light, is just a small portion of all the kinds of light that there are. So if we were to come over here on the right side, we have kinds of light with extremely long wavelengths, sometimes longer than us, things like radio waves, microwaves. And as we go this direction, we hit shorter and shorter wavelengths until we land on X-rays. And it turns out that X-rays do have short enough wavelengths to see the biomolecules. Now, some of you are probably thinking, wait a minute, I've gotten an X-ray done before at my doctor's office or at my dentist's office, and I saw my hand or my teeth, and I didn't even see cells, much less biomolecules. And yes, that's true. But the truth is that X-rays can see individual biomolecules in very special situations. And you think about something like taking an X-ray of your hand, and there's a ton of different biomolecules in there, and they're all on top of each other, and it's almost like the information gets a little washed out. So to think about this in more detail, I like to use a bit of a metaphor. I know some of you guys are in orchestra or band or choir, and believe it or not, biomolecules are kind of like an orchestra. So let's imagine that you're in a big concert hall and you're listening to an orchestra play. You hear a big, rich sound, but you can't really hear the parts of the individual players. So let's pretend that I want to hear the part of this one clarinet player. She's kind of like my biomolecule that I'm interested in. But when they're all playing together, I can't hear her, just the orchestra as a whole. So how do I go about hearing her? Well, the easiest thing you can think of is you just get rid of the rest of the orchestra. But you can imagine that if the concert hall's really big and you're sitting far away, she might be a little too quiet to hear. So how do we hear her? Well, let's pretend we can clone her, and we fill up the stage with clones of this one player, but there's no conductor. And they all start playing her part, but they're playing it all at different times, and it's a little bit overlapping. And in this case, we can get some information. If we didn't know she was playing a clarinet before, we probably can hear well enough to determine that now. We probably can tell if her part is high or low pitched, or we could tell if it's fast or slow. But with it being all overlapping, we can't really hear any details. So let's take this a step further, and let's say that we take our clones and we fill the stage with a perfectly ordered, synchronized assembly of them, and they all start playing at the same time. And when this happens, we can hear her part loud and clear. Now, believe it or not, biomolecules and X-rays are just like this. Let's say that I take one biomolecule and I put it in some X-rays. I probably won't be able to see very much, kind of like I couldn't really hear the one clarinet player by herself. If I make a lot of copies of that molecule and I put them in random orientations in the X-rays, I do get some information, and sometimes depending on the question I'm trying to answer, that might be enough. But I can't really see the details of the structure that way. So if I want to do that, I need to make a perfectly ordered array of molecules and put those in the X-rays, and then I can see the details. So to recap a little bit, the steps to seeing those details of our biomolecules, we first need to identify the molecule of interest. That's like picking out our clarinet player. Then we need to make a lot of just that molecule. That's like our clones. And finally, we need to pattern those molecules to eliminate disorder. Well, that's all well and good, but how on earth am I going to do that? Well, luckily for us, it turns out that nature has a way to pattern molecules perfectly like this. And that way is crystals. So a lot of you are familiar with crystals if you've seen rock candy or table salt, or you might have even seen one of these decorative little gemstones here. And people are always very drawn to crystals because they look very ordered and regular. And that's especially true on a molecular level. If I were to take a saw and chop off part of this crystal, then use my special magical magnifying glass to look at it on a very small scale, I would see that it's made of a perfectly ordered, perfectly repeating array of molecules, and they look the same as far as the eye can see in every single direction. So this is exactly what we want. And believe it or not, you can make crystals out of biomolecules. Here's a picture of an assortment of crystals made up of different kinds of protein. And you can see that they really do look just like little gemstones. I should mention that the size scale is a little bit different. Most of the decorative gemstones you might see might be a couple inches across. But these protein crystals, they might be a 1/16 of an inch or much, much smaller. So they're pretty tiny. But they are crystals. And if you look at them on the molecular scale, we have this repeating array of molecules, which is exactly what we were after in the first place. So this is great, right? I've got my protein crystal, so I'll just, I'll take it down to the dentist office and put it in the X-ray machine and see what it looks like, right? Well, unfortunately, not quite. So it turns out that medical X-ray sources actually aren't powerful enough for this. You can think of your dentist's X-ray source as being a little bit like a light bulb for X-rays. So the X-rays go out and they go out in a broad area, which is really good for looking at something kind of big, like your hand or your tooth. But it's not particularly bright anywhere. And considering we're trying to look at a little bitty protein crystal, most of the X-rays don't hit it and are kind of wasted. So we really want a different kind of X-ray source. We want to replace something like a light bulb with something more like a laser. We would like to see a very bright and focused beam of X-rays about the size of our crystal. Now, luckily for us, this kind of X-ray source does exist. There's something called a synchrotron. Now, a synchrotron is an amazing machine that accelerates subatomic particles like electrons to almost the speed of light. And if you do this in just the right way, they produce a really bright, really focused beam of X-rays. So this is exactly what we want. But you're probably thinking, well, a machine like this, it can't be very common. Where on earth would I get one? And they're not hugely common, but luckily for us, Cornell has its own synchrotron. And if you've ever been on Cornell's campus walking around on the sports fields, you've been on top of the synchrotron, and underneath your feet are little electrons flying around at almost the speed of light. So it's pretty cool, and you can check it out at chess.cornell.edu to learn more. All right, so now that we know about synchrotrons, we can complete our list of how we learn the details of a biomolecular structure. First, we identify our molecule, make a lot of it, pattern it to eliminate the disorder, and then we bring the crystal to the synchrotron to get the structure. Now, I should mention that quite like going to your doctor's office to get an X-ray of your hand. You don't immediately see a picture of the molecule. What you see is an image where you get all sorts of spots, and you do some special image processing and analysis to back the structure out. But this is something that scientists have been working on for a while, and we're actually pretty good at it. And we call this field crystallography. So what has crystallography taught us so far? It turns out actually quite a lot already. So far, scientists have been able to solve the structures of over 144,000 biomolecules, mostly proteins, but there's some nucleic acids too. And even though that sounds like a big number, compared to all the proteins that exist, we're just barely scratching the surface here. So there's still lots more interesting and important science to be done. But the really cool thing about this is all of these structures are conglomerated into this resource called the Protein Databank, which anybody can access. So you guys can Google Protein Databank and see all these structures. In particular, Protein Databank has an educational resource called PDB 101. So you can go there and you can click on different molecules and learn about them and learn about their structure and how they function. There's also some really cool interactive resources in PDB 101. You can make origami proteins or DNA. There's things like coloring books and flyers, so please do check it out. So there's all these structures in the Protein Databank, but I really want to emphasize that this isn't just a scientific curiosity. Crystallography is something that is used all the time in pharmaceutical research to help scientists design better medicines. And there's whole books on the topic if you're interested, and you can check it out. And basically it works just the way we've been talking about, where scientists solve the structure of a biomolecule, and they use that structure to help them design a better medicine that can bind with and interact with that molecule. And in case you're curious, scientists are actually using crystallography to help study the new coronavirus. Just today, I went on the protein databank and I searched for crystal structures from the coronavirus, and I found 135 of them already. So we're working on it. Now, as helpful as crystallography has been to us already, the really exciting thing is that it's still evolving. We're getting better at doing this all the time, and we're able to study increasing numbers of really cool and interesting samples. So on the sample side, we're getting better at isolating and crystallizing more kinds of molecules. We can make crystals inside cells now. We can make crystals in a lipid phase. So we can look at really interesting molecules that we couldn't see before. On the X-ray source side, there's new, even brighter sources of X-rays for scientists to use now. There's something called an X-ray free electron laser, which unfortunately we don't have at Cornell, but here's a picture of one that I've used before at Stanford. And this is an enormous machine, and it makes X-rays that are so bright that we're actually able to watch as proteins function, which is a total game changer. So keep your eyes out for crystallography news, because there's a lot of new cool things that are coming out and will be coming out in the near future. All right, I think that's most of what I wanted to tell you guys today. So just to summarize, today we learned that biomolecules are important little molecular machines that are responsible for most of the functions that happen inside of our bodies. Luckily for us, X-rays have short enough wavelengths to allow us to see biomolecules. And if we make a crystal of our molecule of interest, we can get detailed structural information using these X-rays. These structures can help us understand basic biology, design better medicines, and more. So thank you guys so much for listening. Hope you had a good time and learned something. If you have any questions about crystallography or even about being a graduate student, I'd be happy to try to answer.
Great, thank you so much, Andrea. I learned so much about this. Oh wow, synchrotrons and biomolecules. This is so fascinating. Okay, so my name is Sana. So I'll be asking you some of the questions that our participants have for you. And then since we have about a few minutes, so I'll ask this one question that really stands out. Let's see. So Irene is asking, since bacteria would eventually evolve to be able to resist new types of antibiotics, so would scientists like you have to be constantly creating new antibiotics? And also, if crystallography could be used in helping creating vaccines?
Yeah, those are great questions. So you are right that bacteria are constantly evolving ways to fight off new kinds of antibiotics. And if you were to go to Wikipedia and look at antibiotics, you would see there's several generations of those that scientists have made to try to just stay one step ahead of the bacteria. But it's something that we're still working on, and it's going to be, will always be kind of in this lockstep, and hopefully we can stay a step ahead. In terms of crystallography being useful for the vaccines, yeah, we really hope so. I think that's the reason scientists are working so hard on solving crystal structures of all these different parts of the coronavirus.
Wow, such a useful tool, and then that we were oblivious to it, I guess. Wow, thank you so much.
Oh, thanks for having me.
Okay, let's see. Barry, back to you.
Oh, okay, great. So awesome. Well, thank you. And thanks, Andrea. That was so fascinating. And again, just, I think really relevant to a lot of the stuff that is going on. And of course, everybody's pretty aware of. So it's so cool to see how you can weigh in and interact and help us all get back to our lives and things. So that's really cool to see. So